Now we discuss about the protein. Okay, then remember remember that proteins are the most abundant organic molecule of the living system. Proteins are the most abundant organic molecule of the living system. Proteins form proteins form the fundamental basis of structure and function of life. So it is the it is the most abundant it is the most abundant organic molecules most abundant organic molecules of the living system of the living system. Remember it, living system. Okay, and and it it forms the fundamental fundamental basis of fundamental basis of structure and function function of life. Okay, it is formed it is formed by carbon, hydrogen, oxygen and nitrogen. It is the it is the polymers. It is the polymers of amino acid of amino acids. Okay, in nature there are 300 amino acid 300 amino acid are present in nature. Okay, 300 amino acid are present in nature. Of these only of these only 20 amino acid 20 amino acid takes part in protein formation in protein formation. Remember those informations. Then we discuss about the amino acid, which are the structural unit of protein amino acid. Okay, amino acid are a group of organic compounds containing two functional groups, that is a amino group and a carboxyl group. The amino group is basic the amino group is basic while the carboxyl group is acidic in nature. They are perhaps first molecules formed in the atmosphere of Earth. Remember it. The amino acid are the first molecules formed in the atmosphere of Earth. Okay. So if we draw a structure of amino acid, then it center has a carbon atom which has a hydrogen and a carboxyl group and a NH2 group, means amino group, and has a side chain called R. This this R side chain this R side chain is differ in different amino acid. Okay, so that is the basic structure. So that this part of amino acid is common in all amino acid. Different amino acid have the different has the different R side chain As example, the glycine is the simplest amino acid. The glycine is the simplest amino acid, which has the structure like that: COOH, NH2, and another hydrogen only. Okay, that is the simplest amino acid, named as glycine. Simplest amino acid. So among the 300 amino acid, only 20 amino acid takes part in protein formation or protein synthesis. For that, those 20 amino acid which takes part in protein synthesis are called protein amino acid. So we can classify the amino acid into two groups, that is protein amino acid and non-protein amino acid. The protein amino acid which takes part in protein synthesis and non-protein amino acid does not take part in protein synthesis. So what are the function of the non-protein amino acid? Non-protein amino acid takes part in different purpose, such as some amino acid helps in and some non-protein amino acid act as antibiotic. As example, azathioprine. Azathioprine is a amino acid, non-protein amino acid, which act as an antibiotic, and others are helps in many biosynthetic pathway. Biosynthetic pathway, such as example, ornithine, citrulline. These are takes part in citrulline. They are takes part in urea formation, which occurs inside the liver. Okay, depending on the structure amino acid are classified into 7 groups. Amino acid also classified into 7 groups. These are the following. These are the following. First one, neutral amino acid. First one, neutral amino acid. Neutral amino acid means those amino acid which have one amino group and one carboxyl group. Which have one amino group and one carboxyl group. Okay. As example, glycine, alanine, glycine, alanine, valine, leucine, isoleucine, etc. These are the neutral. The amino acid which have only one amino group and one carboxyl group is called neutral amino acid. Then acidic amino acid. Acidic amino acid means it contains two carboxyl group and one amino group. Two carboxyl group and one amino group. For that they are acidic in nature. For that they are acidic in nature. Acidic in nature. As example of acidic amino acid are aspartic acid, glutamic acid, glutamine, etc. These are the acidic amino acid. Then basic amino acid. Basic amino acid means those amino acid which are contains an extra amino group and an extra amino groups. So those amino acid contains two amino group and a single carboxyl group. As example, arginine. Lysine, etc. These are the basic amino acid. Then sulfur containing amino acid. Sulfur containing amino acid. Sulfur containing amino acid means those amino acid which contains the sulfur in their R side chain. As example, cysteine, methionine, etc. Those are the sulfur containing amino acid. Then alcoholic amino acid. Alcoholic amino acid are those which having a alcoholic or hydroxyl group which contains a hydroxyl group is called alcoholic amino acid. Alcoholic amino acid, as example serine, threonine, tyrosine, etc. Serine, threonine, tyrosine, etc. These are the alcoholic amino acid. Then aromatic amino acid. Aromatic amino acid are those amino acid which possess a cyclic structure or contains a ring, okay, in their side chain. As example, phenylalanine. Phenylalanine is a aromatic amino acid. Tyrosine also has a alcoholic group and also contains a ring. For that, tyrosine are placed into two group, alcoholic and also aromatic group. Okay. Then heterocyclic amino acid. Heterocyclic. Heterocyclic amino acid. Heterocyclic amino acid are those amino acid which have nitrogen in the ring structure. Which have nitrogen in the ring structure. As example, histidine. As example, histidine, proline. Okay, these are the heterocyclic amino acid. So, amino acid depending on the structure, we can classify the amino acid into 7 group: neutral amino acid, acidic amino acid, basic amino acid, sulfur containing amino acid, alcoholic amino acid, aromatic amino acid, and heterocyclic amino acid. Those heterocyclic amino acid also called the amino acid. Sorry. Heterocyclic amino acid are those amino acid which have the nitrogen in their ring structure. Okay. You must remember that proline proline is a proline proline is a amino acid. Proline is a amino acid because they contains imine group NH. We can also classify the amino acid depending on their nutrition, nutritional value. That is, amino acid also classified into 3 groups depending on their nutritional value. These are the essential amino acid, non-essential amino acid, and semi-essential amino acid. Essential amino acid are those amino acid which does not synthesized by our body, by the body of an organism. Those amino acid are obtained from plants through food. Okay, those are called essential amino acid. Means it does not synthesized in body and are often from food, means plant product. Non-essential amino acid are those amino acid which are synthesized by our body, Okay, those are the non-essential amino acid. Semi-essential amino acid, it does not synthesize in a baby, but synthesize in an adult in an adult. So these are called semi-essential amino acid. Okay, then example of the essential amino acid. Essential amino acid are isoleucine, isoleucine, leucine, lysine, isoleucine, lysine, tryptophan, valine, valine, methionine, etc. etc. Isoleucine, leucine, lysine, tryptophan, valine, methionine, etc. Those are the essential amino acid. Okay. Non-essential amino acid, as example, non-essential amino acid is serine, aspartic acid, glutamine, glutamic acid, glycine, tyrosine, etc. Those are the non-essential amino acid. And semi-essential amino acid are, as example of semi-essential amino acid are histidine and arginine. Histidine and arginine are semi-essential amino acid because they does not synthesized in a baby but synthesized in an adult. So those are called semi-essential amino acid. Remember remember some terms such as amphipathic molecule. Amphipathic molecule. Amino acids are also called amphipathic molecule because they have both the carboxyl group, means COOH minus carboxyl group, and have also NH2 group. So in a medium it act as a cation and also act as a anion. For that the amino acid is also called amphipathic molecule. Then another term that is isoelectric pH or isoelectric pH. What is isoelectric pH? In which pH in which pH an amino acid an amino acid balanced by the number of negative charges, number of negative and positive charges okay, in which pH amino acid balanced by the number of negative and positive charges, means it is neutral. It are in It's are in neutral condition. Okay, in which pH the amino acid contains the same positive charge and same negative charge. That pH point is also called isoelectric pH. If we lowers the pH, if we lowers the pH in low pH, if we lowers the pH, then the amino acid shows the acidic in nature, and in high pH, and in high pH it shows the basic nature. Okay, or shows the negative charges, negative charges and in high pH they shows the positive charges. So each amino acid have a specific pH value in which they bears same positive and negative charge. That pH point is called isoelectric pH. Then I discuss about the structure of protein. Structure of protein. The biological function of a protein is due to its conformation. The biological function of a protein is due to its conformation, which is defined as the three-dimensional arrangement of the atoms of a molecule. Four basic structural level are assigned to protein. These are the primary structure, secondary structure, tertiary structure, and quaternary structure. So, a protein has four structures. These are the primary structure, secondary structure, tertiary structure, and quaternary structure. First, I discuss about the primary structure of protein. The primary structure of a protein refers to the number and linear sequence of amino acid in the polypeptide chains and location of disulfide bridges, if there are any. Means, previously I said that the structural unit of protein is amino acid. So the primary structure of protein is formed or composed of the linear structure of amino acid. How does it form? The amino acid, the amino acid are attached with each other by forming a peptide bond. Peptide bond occurs in between the NH2 group, and carboxyl group. NH2 group and carboxyl group. Okay, that is the primary structure of a protein. Then how does the peptide bond is formed? How does the peptide show? That is the peptide bond. These are the peptide bond. Okay, these are the peptide bond. How does the peptide bond forms? The peptide bond the peptide bond forms between the amino group and the carboxylic group of the adjacent amino acid. Look here. That is the simpler structure of a amino acid NH3 plus, then R side chain is present there. That is amino acid 1 and that is amino acid 2. C H C double bond CO COO minus and here NH3 plus. Then it also has a it also has a R side chain, that is amino acid 2. Then how does the peptide bond it form? At the time of peptide formation, they release one molecule of water, and then they attach through a peptide bond. C CO NH CO NH. Here NH3 plus is present and here C double minus is present. The side chain. So that is the peptide bond. CO NH. That is the peptide bond. Peptide bond. Then the peptide bond is formed between the adjacent amino acid and they form a linear structure that is called the polypeptide chain. That is called the polypeptide chain. It forms at the time of protein synthesis in ribosome. It occurs at the time of protein synthesis in ribosome. Okay. So peptide chain is the primary structure of a protein. Then the secondary structure. Next, the secondary structure of protein. How does it form? When the polypeptide chain is formed, it arranged into specific manner that secondary structure also differentiate into three groups. That is, first one alpha helix, second one beta pleated sheet, and third one the collagen helix. Okay. How does the alpha helix is formed? When the peptide chain is formed, the peptide chain is coiled spirally. The polypeptide chain is coiled spirally, generally in right-handed manner. The helix is stabilized by the hydrogen bond between oxygen and carboxylic group of the amino acid. Okay, so alpha helix is the polypeptide chain is coiled spirally, generally in right-handed manner. The helix is stabilized by hydrogen bond between oxygen and carboxylic group of between the amino acid of carboxylic group of an amino acid. Okay, So this structure look like this. Peptide chain is coiled spirally. That is the secondary structure, means alpha helix. Okay, here these are the amino acid. These are the amino acid, and they attach by and forming a hydrogen bond among them. Among them, these are the hydrogen bond. Singly, a hydrogen bond is weaker. But when many hydrogen bond, if many hydrogen bond is present in a structure, then it will be stronger. For that, the peptide chain become a helically arranged structure called alpha helix. Okay, so in alpha helix we can found two types of bond. One is peptide bond, which forms in between two amino acid of the peptide chain, and the hydrogen bond, which occurs in between the oxygen and carboxyl group of amino acid. Then beta plated sheet. Beta plated sheet, how does it occur? Beta plated sheet also classified into two groups. These are first parallel beta plated sheet and anti-parallel and anti-parallel beta plated sheet. In both, many peptide chain takes part. First one, if this is a peptide chain, this is a peptide chain, and this is also a peptide chain. Okay, in beta plated sheet, run in the same direction. Same in parallel beta plated sheet, the peptide chain runs at the same direction, like that, same direction. Okay, then what will form? Then the hydrogen bond will formed in a similar manner. The hydrogen bond will form in a same manner. These are the hydrogen bond. Okay, then in case of anti-parallel, here the polypeptide chain are arranged in opposite direction. Here, then next is that manner, then next is that direction. In case of parallel, they arranged in same direction, but in anti-parallel, they arranged in opposite direction. For that, the hydrogen bond will formed between here. That arrangement of the peptide chain is called antiparallel beta plated sheet. These are the hydrogen bonds. Okay. Then collagen helix. In collagen helix, there are three strand or three polypeptide chain coiled. around one another. Three polypeptide chain coiled around one another means in collagen helix that is a peptide chain, that is also a peptide chain, and that is also a peptide chain. Okay, so here three peptide chain takes part, here three peptide chain takes part. Okay, that is the collagen helix. Then the next means tertiary structure. Tertiary structure. In tertiary structure, the helical polypeptide molecule means the secondary structure may fold on itself and assume a complex but specific form like spherical, rod-like, or any form of between these. These geometrical shapes are also known as the tertiary structure of protein molecules. Okay, then here what happen? Suppose this is a helix, that is a helix, and also that is a another helix, that is another helix, and here those helix are attached by forming different kinds of bond, different kinds of bond, and they form the tertiary structure of a protein. Okay, so what are the bond which are takes part in formation of the tertiary structure of a protein? Those bonds are hydrogen bond, hydrogen bond, ionic bond, ionic bond, hydrophobic bond, hydrophobic bond, covalent bond, covalent bond, etc. Those bonds are takes part in the formation of tertiary structure of a protein. And then the last means the quaternary structure. The quaternary structure, quaternary structure of protein. What happens at that case? Here the many tertiary structures are get together by different kinds of bond. Okay, proteins are said to possess quaternary structure if they consist of two or more polypeptide chain united by forces other than covalent bond. Okay, so here peptide bond and disulfide bond does not occur. Without peptide and disulfide bond, another bond are formed such as hydrogen bond, electrostatic bond, etc. Okay, those bond are takes part in the formation of quaternary structure. How does it occur? Suppose that is the tertiary structure, means those are the peptide chains. Those are the peptide chains. Those peptide chains are attached by different kinds of bond. to form a specific protein. Here only covalent bond and peptide bond does not occur. Other bonds will form. Okay. So the protein get the final structure. So the protein gets the final structure. Then I discuss about the different kinds of bond. Different kinds of bond occurs in or helps in protein synthesis or protein formation. That is first one, ionic bond. First one, ionic bond. What is ionic bond? Ionic bond, it occurs to attractive force. It occurs due to attractive force between oppositely charged ionized group. It occurs due to attractive force due to attractive due to attractive force between oppositely charged ionic groups. As example, the NH3 and the bond formed between the NH3 plus and C double OH minus. So peptide bond is also a ionic bond. Then the hydrogen bond. Hydrogen bond. Hydrogen bond develops due to the sharing of hydrogen. It occurs during the sharing of hydrogen by 2 electronegative atom. Electronegative atom. Okay. Then the covalent bond. Covalent bond. What is the covalent bond? Covalent bond are the strongest. Covalent bonds are the strongest bond between the different kinds of bond. There are two types, peptide bond and disulfide bond. Peptide bond is formed when a water molecule is eliminated. Peptide bond is formed when a water molecule is eliminated during a reaction between the amino group and the carboxyl group of the amino acid. And disulfide bond may be formed between different chains of amino acid which contains the sulfate, which contains the sulfur in their R side chain. If any amino acid contains like cysteine, if any amino acid like cysteine contains the sulfur, then among those amino acid the disulfide bond will occur. That is the disulfide bond. If sulfur is present, then only the disulfide bond is occur. So disulfide bond and peptide bond is a type of covalent bond. Next, the hydrophobic bond. Hydrophobic bond. What is hydrophobic bond? It is formed between two non-polar group. Okay. It helps in excluding water in that area and increasing compaction. It helps in excluding water in that area and increasing the compaction. That is called hydrophobic bond. So hydrophobic bond formed is formed between two nonpolar nonpolar groups by excluding water. Okay, those are the different kinds of bond which takes part into the formation of protein structure. Next, the classification of protein. We can classify the protein into three groups. These are the simple protein, conjugated protein, and the derived protein. Simple protein are those which are composed of only amino acid. Conjugated protein are those amino acid when it contains a non-protein part. Means if a protein attach with the when a protein is formed by the amino acid and other substances, then these protein are called conjugated protein and derived protein. These are the denatured or degraded product of the simple or conjugated protein. The products formed by the degradation or denaturation of the simple or conjugated protein. Those products are called derived. Then simple protein also classified into two groups. These are the globular protein and fibrous protein. Globular protein, these are the spherical or oval in shape. And fibrous protein are like filamentous, okay, or fiber like in shape. So globular is round or oval in shape and the fibrous is fiber like shape. Okay, these are the fibrous protein. As example of globular protein, albumin which are present in egg, globulin protein are present in hemoglobin, glutathione are present in grains. Okay, glutathione are present in grains. Histone is a nucleoprotein. Which helps in the packaging of the DNA. And the fibrous simple protein, as example, collagen fiber which are present in the connective tissue of animal. Elastin fiber, it is also present in yellow fibrous tissue. Collagen fiber is present in white fibrous connective tissue. Okay. Keratin also present in our hair, nail, etc. Keratin protein. Those are the fibrous protein. Then Conjugated protein, amino acid and other substances are attached to form those protein, such as nucleoprotein, nucleoprotein if the protein attached with the nucleic acid, then glycoprotein if the protein conjugated with the glucose or carbohydrate. Then lipoprotein means if the protein contains or attached with the amino acid attached with the lipoprotein. It also occurs in plasma membrane. Lipoprotein. Phosphoprotein if proteins has the phosphate group, then these are called phosphoprotein, etc. etc. Those are the conjugated protein. As example of derived protein, such as coagulated protein, okay, coagulated protein, fibrin protein which are present in our blood plasma, helps in blood clotting. Those are the derived protein. Okay, we can classify the derived protein into two groups. These are the first one, primary derived protein, and another is secondary derived protein. As example of primary derived protein are coagulated protein. What are the coagulated protein? These are the denatured protein. Produces by the agent such as heat, acid, alkaline, etc. Coagulated protein. Then metaprotein. These are the second stage product of protein hydrolysis obtained by treatment with the slightly stronger acid and alkaline. Okay, so those are called metaprotein. And secondary derived protein are those which are progressive hydrolytic product of protein hydrolysis. Okay, as example proteases, then polypeptides, peptides are the secondary derived protein.